today we will discuss about the protoplast fusion. Okay, so this protoplast fusion comes under the plant tissue culture. Okay, so why we use the plant tissue culture? Because we need the different variation of the plants. Okay, means we wanted to some resistant type of plants, some more producing fruit plants, some producing those plants which can survive on all kind of seasons. Okay, so like for that we require the plant tissue culture. Okay, but in plant tissue culture, there are certain hurdles we can't do it by the natural condition. Okay, so what are those limitations? The breeding is sexual, there is sexual incompatibility. Okay, in the plants, so that's why we can't breed one plant to another plant. Okay, through the natural process. So but through plant tissue culture we can do that thing also. So that is the advantage of using the plant tissue culture. And in this process, protoplast fusion is one of the most important mechanism. Okay, through that we can grow the variety of multiple plants. Okay, so the another thing is the limitation of the producing different type of plant is the gene transfer into plant, because there is the presence of cell wall. Okay, so as you all know that plant cell is having this is suppose plant cell and this is the animal cell. Okay, so what is both are belongs to the eukaryotic cell, right? But there is certain differences. The more means the basic difference is it is having the plant cell is having the cell wall as a outer layer. Okay, it is having the cell wall as a outer layer, which is absent in case of the animal cell. So some modification is easier in case of the animal cell, but in case of the plant cell, because of the presence of cell wall, it is difficult. Okay, so the first thing is the sexual incompatibility. That's why we can't grow the multiple variety of plants. And second thing is the presence of cell wall. Okay, so that's why we can't directly easily transfer the gene of one plant to another plant. Okay, and yeah, so the production of hybrid plant uh, if you wanted to some property of one plant to into the another plant, then this kind of plant is called as the hybrid plant, because that characteristic not belong to its natural parents. Okay, it borrows from the other plant. So that's why the production of hybrid plant through the fusion of two different plant protoplast is known as the somatic hybridization, and such hybrid is called as the somatic hybrid. Okay, so suppose this is one plant and this is the another plant, but this plant is having some characteristic and this plant is having some other characteristic, but you wanted that your hybrid should have the characteristic of both the plants. Okay, so what you will do in this case is, because presence of the cell wall, it is not possible to directly fuse these two cells. Okay, that's why we are isolating the protoplast, which is in that condition, protoplast means it is in that case the cell wall is absent. Okay, so for cell wall is absent, and when you fuse these two protoplast of the two plants, then whatever hybrid will create, that hybrid is called as the somatic hybrid, and this process is called as the somatic hybridization. Okay, so this somatic hybridization is very much important to produce variety of the plants in a large number of in the large number. Okay, so that's why in plant tissue culture we use the somatic hybridization. Somatic hybridization is nothing but, somatic means some means rest of the, it is not the germ cell, it is the somatic cell which means make our body. Okay, or the plant body. So that somatic cell, when you will take and fuse the protoplast of that two cell of two different plants, that process is called as the somatic hybridization, and whatever hybrid is produced, that is called as the somatic hybrid. Okay, so and protoplast means here is the whole content of the cell apart from the its cell wall. Okay, so that is called as the protoplast. So this is the procedure how you will generate the hybrid plant through the protoplast fusion. Okay, so firstly you have to isolate the protoplast. Then after isolating this protoplast, you have to fuse the protoplast of desired species. Okay, means suppose you wanted two things. One is your plant should be resistant to like many insect. Okay, and another is the you want this plant should grow in every season. Okay, so there is two type of plant. Okay, so this is two desired characteristic which you wanted. Okay, but each plant is having only one characteristic, so you wanted to produce the hybrid which is having both the characteristic. So that's why you wanted to fuse your protoplast of desired species, means means between which species you wanted to do the fusion. Okay, so that is called as the second step, which is the fusion of protoplast of desired species. Okay, then third step is the identification and selection of the somatic hybrid cell. Okay, so like after fusion there is not necessarily like all the fusion will give you the hybrid cell, right? Some will be not fused even, and some will be fused but not in your desired with the not desired cells. Okay, means this two type of cell can fuse together, means among themselves, and this type of population also can fuse together. Okay, and some of them in the population will be not fused. Okay, so the identification and selection of somatic hybrid cell will be very much essential, because not all in the population not all the cell will be your hybrid cell. Okay, your desired hybrid cell. Okay, so for that you need the identification selection. Then you require the culture of the hybrid cell. Like once you isolated your desired hybrid cell from the population, you wanted to means you have to culture it to obtain in more number, right? So for that culture of the hybrid cell is essential. And after that like you this cell should be grow as a plant. Okay, so for that it require the regeneration of the hybrid plant, right? It should be having the capability to produce all kind of like it should produce the leaves, it should be produce the shoot, and it should be produce the root. All this regeneration should be happen in the hybrid plants. Okay, so that is the whole procedure how we do the somatic hybridization. Okay, so the first step means we will see in the very detailed manner like how this all step is happen. So first one is the isolation of the protoplast. Okay, because presence of the cell wall you can't directly fuse two plant cell together, so that's why we require the protoplast. Okay, so the isolated protoplast is highly fragile and the outer membrane is fully exposed. And yeah, so directly I can go like what is the method of the isolation of protoplast. So there is two basic protocol. Okay, so first one is the mechanical method, and these two are the enzymatic method. Okay, this is the enzymatic based method, and this is the mechanical method or non-enzymatic method. Okay, so for any kind of fusion firstly you require the protoplast. Okay, so in the mechanical method, here in the graph you can see firstly you have to take the leaf. Okay, in the leaf there is epidermis and epidermal layer, and then you are having the cell wall. Okay, so this leaf cell you have to firstly chop down, okay, into small pieces, and after that you have to put this leaves into the osmolytic solution. Okay, so in that case what will happen is when you add it into the osmolytic solution, it will um, uh, through the osmotic process the cell wall will be burst. Okay, and your whatever content will be inside your cell, it will come out, okay, in the solution. Okay, then what you have to do is you have to just mechanically this sorry you have to mechanically dissect your cell, okay, and then isolate your protoplast to protoplast will be released. When you means mechanically means whatever left which is not burst or which is not come out because of the plasmolysis solution, then through the mechanical step you can means by manual method you can take out all those things, and this is your protoplast which will be released, okay. So then second thing, this is all about your mechanical method, okay. Now you are having the sequential method and simultaneous method. It is nothing but both the method is belongs to the enzymatic method, okay. So in the sequential method, what we do is the tissue is first treated with the pectinase enzyme, okay, and what this enzyme will do is this will separate cell by degrading the middle lamina, okay. So suppose this is the leaf, okay. So leaf is made up of means millions to billions cells, okay. So the cells will be separated by this pectinase enzyme because it will degrade the lamina, okay. Lamina is connective tissue, okay. We can say that it is the connective tissue, connective material which will help into binding of one cell to another cell, okay. So it will degrade this lamella and this pectinase, then you these cells will be free, okay. And after freeing these cells, what will happen is you have to dissolve the cellulose because your cell wall is made up of the cellulose. So this cellulose to this cellulose will be break by cellulase enzyme, okay. So then this cell wall will be also dissolved, okay. And this your protoplast will be released, okay. This is the sequential method because one by one we are adding these enzymes, okay. But in case of the simultaneous method, what is happening is we are adding both the enzyme together, okay. So we are using the mixture of pectinase and the cellulase enzyme, okay. And most of the case we use this simultaneous method because it will save our time, okay. And that's why we use this simultaneous method more, okay, over the sequential method. But in both the method, the most common method is the enzymatic method because it will give you the good yield of the viable cells, and there is no damage of the protoplast. Because when you are using the mechanical method, you have to through needle you have to just like separate the cell wall things or the left out things with the needle, okay. So that time your protoplast can be degraded, okay, because of the needle. So that's why the enzymatic method is more safe comparative to the mechanical method, okay. Yeah. So in the enzymatic treatment, the important factor is the this pectinase will degrade the middle lamella and cellulose will digest the cell wall, okay. And there is another enzyme, but this is not that much essential. These two are the major essential enzymes, but apart from these two, there is another enzyme, that is the hemicellulose, which digests some of the tissues, okay. So suppose there is like a very hard cell wall is present, the cellulose content is more, that time we used extra hemicellulose enzyme which will digest the some tissues, okay. And the pH means while performing this enzymatic reaction, the pH should be always 5 to 8, okay. And the temperature should be 25 to 30 degrees Celsius, and the time is taking is 30 minutes for 30 minutes to 1 hour suppose, okay. So this is the ideal condition to work these enzymes to properly degrade the cell wall and the middle lamella, okay. And this osmoticum is maintained by the sorbitol or mannitol. This is the carbohydrates, okay. So these are the major things you require to isolate the protoplast, okay. Then in the second step you have to fuse the two protoplast, okay. So the plant protoplast represent the finest single cell system that could offer exciting possibility in the field of somatic hybridization. Okay, so in this case, what you have to do is either mechanical method or either induced method, or I can say like a spontaneous method. You have to fuse two protoplast, and this is your, you can see here, this is the tobacco plant protoplast. Okay, two tobacco plant protoplast. Okay, so it is obtained by the digesting away the cell wall. Then you fused this, means either you put it into the any kind of inducing solution, or by mechanically when you fuse this cell, it will create the somatic hybrid, which is having the characteristic of both genetic background, both the cell genetic background, and it can be regenerated into the plant with some trait of both parental plants. Okay, so here in because both of the nucleus is present in this cell, so that's why it will develop the plant which is having the trait of both of the parental plants. Okay, so after that you have to do the protoplast fusion. Okay, this protoplast fusion can be used to make crosses within species, like it is if it is happening within species, it is called as the intraspecific, because it is happening in inside of the species, that's why it is intraspecific. When it is happening between species, then it is called as the interspecific, and within genera it is called as the intragenic, and between genera it is called as the intergeneric. Okay, so these four type of crosses you can make by the protoplast fusion. So there is three different method of protoplast fusion. Okay, so the first one is the spontaneous method, second one is the mechanical method, and third one is the induced method. Okay, so in the spontaneous method, when two protoplast will keep together, then the adjacent protoplast in enzymatic mixture have the tendency to fuse together. Okay, so to form the either homokaryon or yeah, in case of spontaneous fusion, there is the more chances of homokaryon production, because two cells is having the capability, or two cells of the same species is having capability to fuse together. Okay, so this type of fusion is called as the spontaneous fusion. Here you are not having any kind of control. Okay, means you can't do any kind of manipulation, because it is happening spontaneous. Okay, and this protoplast is fused through their plasmodesmata. Okay, and firstly through plasmodesmata it will adhere to each other, and then it will fuse. Okay, and this spontaneous fusion is of no importance for the somatic hybridization, because here you are not having any control, means it will fuse automatically, right? So you can't create the desired property in this case. So that's why spontaneous fusion is not generally used in case of the somatic hybridization. Okay, second thing is the mechanical fusion. Okay, or you can say like it is the physical fusion of protoplast under the microscope by using the micro manipulator and perfusion micro pipette. Okay, so there is one needle type of instrument is called as the micro manipulator. Okay, so what you have to through that, what we can do is, we can after isolating the protoplast, we have to just means by mechanical force we have to just fuse these two cells. Okay, and this fusion doesn't need any kind of fusion inducing agent, because here the needle is we are forcing we are applying some force to fusion. Okay, so that's why it is called as the mechanical fusion, but it is not advisable, because it can like disturb your protoplast. Okay, like in case of the isolation of protoplast, there also we have seen like mechanical isolation. So in that case, in this case also we are doing this thing by the sharp needle. Okay, so that's why it can harm also our protoplast. Okay, so that's why it is also dangerous. Okay, but we can also do it by mechanical fusion also. Okay, And the third one, which is most used fusion, is the induced fusion. So in the induced fusion, what we do generally do is we isolated the protoplast, and we will keep into the inducing chemicals. Okay, we will deep this two protoplast into the inducing chemical solution. That chemical is called as the fusogens. Okay, because it is helping in the fusion of two protoplasts, so that's why it is called as the fusogens. And the example you have to remember, like in exam point of view, it can be asked like what are the fusogens which will useful for the protoplast fusion. Okay, so that is the most common example is the sodium nitrate and PEG. Okay, these two, and calcium as well. It is these three fusogens are most important and most commonly used fusogens. Okay, or it is helpful in the protoplast fusion. Apart from that, high pH require, as we have seen in the there is 5 to 8 pH should be there, but for the for the isolation it require 5 to 8 pH, but for the fusion it require more than that. Okay, in the basic condition the fusion will be very good. Okay, and then your polyvinyl alcohol also work as a protoplast fusing agent. Okay, and artificial sea water, because it is salty in nature. Okay, so it will also help in the fusing two protoplast. Okay, this chemical fusion cause the isolated protoplast to adhere each other and lead to the tight agglutination followed by fusion of the protoplast. Okay, so when you dip these two protoplast into the chemical solution, then it will help to adhere these two protoplast together. Okay, and after that it will be agglutinated and fused. Okay, and this is very means most common, or non means most common fusing process. Okay, so induced fusion we generally used in our laboratory. Okay, so yeah, this is the mechanical fusion. Sorry, the fusion process, how fusion process is happened. Okay, so firstly the addition is happened. Okay, so when you treat the fusion solution, these two protoplast, okay, then it will come together and adhere to each other. Okay, here you can see. Okay, and after certain time, through some kind of chemical reaction, the plasma membrane will open, or it will fuse together. Okay, and the plasma membrane of two cell will be fused together, and it will create this cytoplasmic bridge. Okay, so this is the cytoplasmic bridge when there is no cytoplasm in the intersecting position. Okay, there is no cytoplasm, so that position is called as the cytoplasmic bridge. And after that these two nucleus will form a single layer of the plasma membrane, and create the hybrid cell. Okay, and this hybrid cell is called as the somatic hybrid, where the cytoplasm as well as the nucleus of both the cell is present. Okay, so this can lead to either formation of this hybrid is called as the homokaryon as well as the heterokaryon. Okay, when you are fusing two same cell, then it will lead to the homokaryon, and when you are fusing two different type of cell, then it will lead to the heterokaryon. Okay, this hybrid is called as the heterokaryon, and if you are fusing two same species cell, then it is called as the homokaryon. Okay, so firstly the addition will occur. Okay, and after that there will be fusion of plasma membrane of both the cell, and then cytoplasmic bridge will form and fusion will occur. Okay, and after some time it will take the shape of the original cell. Okay, Then the third step is the identification and selection. So once isolated the protoplast, then fuse the protoplast. Now you have to identify and select those protoplast which is having your desired characteristic. Okay, so for that what you have to do is, there is different method to identify those protoplast. Okay, so it can be based on the morphological basis, it can be genetic and physicochemical physiological complementation, and through certain dye also you can identify those means hybrid cells. Okay, suppose in case of the morphological basis means, you know that the hybrid cell will be what type of possessing what type of shape. Okay, so based on suppose this round shape is your desired shape. Okay, and there is this kind of shape will be there, and this type of cell is there. So by looking into the different type of cell through the microscope, you can identify like which is your hybrid cell. Okay, but through morphological basis you can't exactly identify those thing, but this is we can use also. Okay, the second thing is the genetic approach. Okay, so through isolating the DNA you can do the PCR and all thing, and you can find out that in which cell is having your desired gene sequence, or which properties having in different type of hybrid cell. Okay, then another approach is the fluorescent label. Suppose you label this cell in with the some green color fluorescent, and this you suppose labeled with the red color fluorescent. Okay, so what then it will give you you know that after fusing these two color, it will give you the yellow color. Okay, so by looking into the yellow color you can say that this is my desired hybrid cell. Okay, so this is the most common or most positive approach to find out the or selecting the protoplast. Okay, hybrid protoplast or somatic hybrid. Okay, so yeah, then you have to, after isolating the protoplast, you need to culture this protoplast. Okay, so in the first step in the protoplast culture is to develop a cell wall around the membrane of isolated protoplast, because at the end you require the original shape or original constitute of the cell, of the plant cell. Okay, so that's why culturing of protoplast in the culturing of protoplast, what will happen is cell wall around the membrane of the isolated protoplast will be developed. Okay, and then this is followed by the induction of division in the protoplast, derived new cell giving rise to a small cell colony. Okay, so suppose after generation of the cell wall outside of this cell, now it required those capability to divide. Okay, then only, because it is only single cell, right? So you need the bunch of the that particular cell, then only it can be grow as a tissue, grow as a organ, right? So that's why this is followed, it should be having the capability of division. Okay, so for that the protoplast derived new cell giving rise to a small cell colony, then only it can divide and produce the its large number. Okay, and then this isolated protoplast or their hybrid cell are cultured either in the liquid or agar media. Okay, so the common practice of using a liquid culture medium include, because it is the suspension kind of culture. Okay, liquid is the suspension kind of culture, so there will be more aeration will available in case of the cell growth. Okay, so that's why we generally prefer the liquid media when you are growing the primary culture. Okay, so not the agar media, but in certain cases we also prefer the agar media because it is providing the stable surface to grow. Okay, but in case of the liquid there is more agitation, more aeration, so that it can grow fast. Okay, so The common practice of using a liquid culture media include either incubating protoplast or heterokaryon in a thin layer or a small drop of the nutrient medium inside a petri plate, which it means when you kept on the particular temperature or light, low light and dark condition at 25 to 28 degrees Celsius, then it will grow as a callus form. Okay, so or it will create a certain bunch of the cell. Okay, so this is the culture of protoplast. Now in the final step you require the protoplast regeneration. Okay, so uh, this is like this process is regenerated from the this from the culture of the protoplast, means this protoplast regeneration comes under the culture of protoplast only. Okay, here also the cell wall will arrange. Okay, means firstly the cell wall created, now it will arrange into the proper shape, and it is means make other constitute of the cell also. Okay, and develop as a callus, or after some time it will create a whole plant. Okay, means when you put your callus tissue into either agar plate or into the liquid media, then it will after some time it will firstly the shoot will regenerate, and then after that root will create, then you have to keep into the field, and then it will grow as a whole plant. Okay, so this is the condition like how this protoplast will regenerated. Okay, 2 to 3 weeks it will take, and after that this callus will grow as a whole plant. Okay, so this protoplast regeneration comes under the culture of protoplast. Okay, so this culture of protoplast is the final step, and protoplast regeneration means here your protoplast will be dividing into different not different cell, means dividing into its progeny, so that it can produce the multiple cells. Okay, so multiple cell will create the tissue, and this more tissue will create the organ, like that finally it will attain the whole plant. Okay, so this is called as the protoplast regeneration. Here only you do the culture of the protoplast. Okay, till the callus. Okay, so here you means to multiplying the cells, here you are doing the culture. Okay, and after that you have to regenerate this protoplast by using different type of hormone or chemicals, like that. Okay, so when you want um, um, shoot generation, root generation, there is different type of chemical we used in the plant tissue culture, like cytokinin, auxin, like that hormone we have to put into the liquid media. Okay, then it will induce the generation of shoot and root. Okay, so this thing here we have to do. Okay, so this is the step of the whole somatic hybridization. Okay, so this is called as the protoplast. The another thing of the somatic hybridization is the hybrid. Okay, here in this case of the protoplast fusion, we have seen the somatic hybrid. Okay, somatic hybrid is having the nucleus of both the cells and as well as the cytoplasm of both the cells. But in case of the hybrid, what is happening is the nucleus is derived from only one parent, and the cytoplasm is derived from the both the parent, is referred to as the hybrid. Okay, and this genetically hybrid are hybrid only for cytoplasmic trait. Okay, suppose you wanted the cytoplasmic characteristic of other cell, but not the nucleatic characteristic or genetic characteristic. Okay, you just wanted their cytoplasmic characteristics, so in that case you require to produce the hybrid. Okay, so in case of hybrid, here you can see that this is two protoplast. Okay, when they fuse together, that is called as the heterokaryons. Okay, and these two heterokaryon, this is one heterokaryon and another heterokaryon, when fused together there is two possibility. Either like when you fuse together, A and B will cell will fuse and it will create the somatic hybrid, which is having the cytoplasm as well as the nucleus of both the cells. But in case of the hybrid, what you have to do is, from the heterokaryon one nucleus you have to means remove, either through the radiation or through the you have to do it as a enucleated condition. Okay, one cell is not cell, this heterokaryon will be not having, sorry, in case of the cell itself, it is not should not having the cell, sorry, nucleus, and one cell is having the nucleus as well as the cytoplasm. Okay, so when you fuse these two type of cell together, then it will give you this kind of cell which is having cytoplasm of both the cells and nucleus of only one parental cell. Okay, so that is called as the hybrid. Okay, so this is the basic difference, but the fusion and the growth of the plant will be remain same only, like we have seen for the somatic hybrid. Like that, hybrid also will work like that only. Okay, the only difference is means what kind of changes you want. If you wanted the whole characteristic, cytoplasmic as well as the means genetic characteristic, so you require to fuse the complete cell material of one to another. But in case of only you require the cytoplasmic trait, then you have to just enucleated one cell and fuse this enucleated cell to another cell which is having the cytoplasm as well as the nucleus, and then it will give you the hybrid. Okay, but the procedure of how to isolate the protoplast, how to fuse the hybrid, means protoplast, then selection of the hybrid, and then culture of the hybrid, both all the thing will be remain same only. Okay, just the mechanism of fusion is different. Like in case of the somatic hybrid, here you are fusing the complete two cells, but here in case of the hybrid, you are fusing the cytoplasmic cell which is not having any nucleus to one cell which is having the nucleus plus cytoplasm. Okay, okay, so there is another question. If the protoplast are placed in the distilled water, they swell and burst as a result of endoosmosis. The plot representing the kinetics of burst is, what will be the correct plot? Then A will be the correct answer. Why? Because here this plot is between the, in the x-axis there is time, and here it is the number of intact protoplast. Like, so in any population there will be certain number of cells only, right, or protoplast only. Okay, so it can't be continuously, means means once you add it into the distilled water, it directly not will means bursting, right, it will take some time. Okay, so here this is the time it is taking, and after that bursting, the cell the protoplast number will be decreased, and at a point the protoplast number will not further decrease, because it is having the only particular number of cell, right, or protoplast. So that's why it won't be like the graph won't be like this type. It is what it is saying is, the intact cell is after placing itself into the distilled water, it is continuously decreasing, and there is infinite decrease. Okay, but in real case, when you put your cells, or when you put your protoplast into the distilled water, suppose you have given a certain number of protoplast only here, right, so for bursting itself it will take some time. So that is the like adapting period, and after that it will start bursting, and in this point there will be no cell left to burst, right.